Hi, good evening, everyone. We welcome you once again to our webinar session. Today's speaker is our very own Dr. Stephen Fleming, who has previously presented for us. And he is the Director of Embryology at Cooper Surgical Fertility Solutions. He has established the world's first master's degree in ART and has been an honorary associate at the University of Sydney. He is an author and editor and has published a number of books, book chapters, and reviewed various journals and articles. And his main research interests include cryopreservation, a topic on which he is going to speak today. So he's going to tell us about the very latest and emerging technique, is the ultra fast warming of vitrified blastocysts. It People have claimed and studies have claimed that this technique is going to help us minimize the embryologist workload, the time, and also give better results. So let us all hear today what Dr. Stephen has to say about this technique. And with this, I hand over the virtual mic to you, sir.
Thank you very much, Preeta.
You please share your screen now, and we welcome you to the session.
I'll share my screen. There we go. Oops. Sorry, try that again. There we go. Okay, so hopefully you can all see that.
Yes, we can.
Good. Is that out of the way? Better. Okay, wonderful. Well, thank you once again, Indira IVF and the organisers, for inviting me along to talk to you all today. As you can see, I'm going to be talking about superfast warming of vitrified blastocysts, but I should also talk a little bit about recovery culture of vitrified blastocysts, and also a little bit about superfast vitrification of blastocysts as well. So hopefully we can cover everything that should be of interest to you today. So to put recent innovations into perspective, it's worthwhile recapitulating the essentials for success with traditional vitrification and warming, since these, of course, will apply equally to superfast vitrification and warming. So when implementing a new technique, we hope we're going to get really good outcomes. However, to get consistently good results, we should be precise and follow very clear and well-established rules. And it covers a lot of these factors here. So first and foremost, the quality of your oocytes and embryos are going to, of course, be the major determinants of success. And then all of these other factors, so the choice of the cryoprotective agents, the temperature, volumes of media, timings, and whether you collapse blastocysts, for example, or carriers you're using, these will all be implemented in your clinic, and you need to make sure that those variations are followed religiously by all practitioners. So, of course, usually protocols are designed to work at either room temperature or 37 degrees C. However, you have to bear in mind what room temperature means in your laboratory. So, of course, the speed of the process is going to be faster. Let's say if your lab is around the sort of upper 20s, 28 to 30 degrees, than if you're managing to control the temperature of your lab in that sort of typically 20 to 24 degree C range. Volumes are really important. So larger volumes may reduce the effects of any errors that you may have, especially when starting up these techniques. And of course, if you're using very small volumes, you run the increased risk of evaporation and therefore increased osmolality of the solution. One of the good things, of course, with fast vitrification is that you are minimizing that time of exposure to these vitrification solutions. But generally, the take-home message is it's always better to start with larger volumes first to ensure that you're getting good results. And then, of course, the operator skill or expertise that will increase with time and experience, but it's really important, again, that you continue to monitor that amongst all practitioners. So if we look at the different vitrification systems, then typically propane diol in combination with dimethyl sulfoxide or ethylene glycol, they're going to be the main cryoprotective agents of vitrification kits, and they all do the same job. And this is really what we're talking about today, and that's driving the dehydration and rehydration processes in the cells. So despite a number of differences in the pH buffers that are used, the supplements, and slight differences in terms of the protocols, all of these kits have happy users and can be optimized for your individual lab settings. And in fact, there's no clear evidence in favor of one vitrification or cryoprotectant system over another. But of course, they can all be detrimental if you don't stick to the times and the temperatures and use them properly. And what's become apparent, certainly since this early study in 2018, a group from Italy, Loda Parmigiani et al., they published a study that assessed the clinical efficacy and efficiency of so-called off-label universal warming. So this is where, rather than using the same warming kit with the same cooling kit, you can mix and match if you have to. And they looked at this on vitrified human blastocysts and analyzed their cryosurvival and implantation rates. And you can see that in this prospective randomized controlled trial on 315 blastocysts, each embryo being randomized for vitrification using, in this case, 2 different vitrification kits, Sage and Kitazato. And then at warming, again, they were randomly allocated to either Kitazato or Sage warming kits. And you can see that the female mean age, cryosurvival rate, these were, and the implantation rates were all statistically comparable between groups. So cryosurvival rates are high, as you would expect, 98.4 to 99%, and implantation rates 18 to 22.5%. Now, one of the things that I'm going to be talking about is this concept of determining your optimal timings, and this has been historically done by observing the mean time, for example, for oocytes to and embryos, of course, but in this case for oocytes to fully re-expand. So that means at least 90% of the original volume after placement in equilibration solutions. So initially, of course, you're going to get you're going to get dehydration of the cells, so shrinkage of the oocyte as the water passes out faster than the permeable cryoprotectants enter the cell. And then, of course, once you've established your mean time of equilibration, you then incorporate that into your standard operating practice within your laboratory. But of course, these timings historically have been stage dependent because of the relationship between the surface area to the volume of different cell types. Of course, oocytes very large, whereas cells within the blastocysts, for example, very small. And also, of course, these timings tended to be reduced if you collapse the blastocele, because the permeating cryoprotectants didn't have to permeate across the fluid within the blastocele. And also the idea that gradual addition of cryoprotectants to oocytes may be advantageous. So this was the historical landscape, if you like, of what we've understood in terms of achieving cryosurvival and competency of these oocytes and embryos to carry on developing. So when it came to warming, we knew that that was really important. So the idea was that you pre-warm both the media and the warming dish in advance, well in advance, so you know that they're going to be at 37 degrees when you take them out of the incubator. And then to ensure that the temperature at 37 degrees is maintained by validating your heat stage over that 1 minute warming period. And then moving the carrier from liquid nitrogen into that warming solution as quickly as possible, again, to increase the warming rate. And although publications often included some discussion of the possible contribution of the warming rate, there's only been actually very few experimental attempts to separate the relative importance of each step. And one of these, Secchi and Mazur, using the mouse oocyte model, showed the importance of the warming rate in their experiments. So you can see that with the highest warming rate of 2,950 degrees C per minute, in those boxes, you get the very highest cryosurvival rates, pretty much irrespective of the cooling rate. So this concept gave rise to the predominance, if you like, of the warming rate over the cooling rate. So that was great, and vitrification was certainly a paradigm shift over slow freezing and thawing. But then there came another paradigm shift in our understanding of what is actually happening during vitrification. And this was the Gallardo study. So in this study, they used in silico, which is a MATLAB program using 2-parameter permeability models, and also in vitro observations of oocyte and zygote osmotic behavior to indicate that dehydration upon exposure to standard cryoprotectant solutions actually occurs very quickly. And the point of minimum volume of the shrink swell curve that you can see, so the not the dotted line, but the solid line shows you the swell shrink curve, and you can see for short dehydration on the left and for standard dehydration on the right, you can see that those minimum volumes are the lowest point of that curve is reached actually very quickly within 60 seconds. And at that point, intracellular water ejection is therefore complete. And that, coupled with the permeation of low molecular weight cryoprotectants, actually results in similar intracellular and extracellular solute concentrations. And you can see those solute concentrations in the dotted line. So, you start off with obviously lower concentrations with the equilibration solution and higher with the vitrification solution. So since those are achieved, then there's prolonging the exposure to cryoprotectant solutions will not have any further impact on the cytosolic glass forming tendency of the vitrification solutions. So in fact, 2 consecutive exposures of 1 minute to 2 standard cryoprotectant solutions, so typically the 7.5% and then the 15% plus the sucrose at 0.5 molar, was sufficient to attain the critical intracellular solute concentration necessary for successful vitrification. And they confirmed this by looking at post-warming cryosurvival and initiation of cytokinesis. And they concluded that, in fact, human oocytes and zygotes are ready to vitrify after 2 minutes. So if you look at the superimposition of the 2 graphs for the standard and the ultra-fast warming, you can see that those solid lines coincide. In fact, it's after 29.4 seconds, so before 1 minute, you get this minimum volume of 44.8% of the total volume. And with the standard protocol of 10 minutes of equilibration, 1 minute of vitrification, um, then you get it re-expansion back to about 79.4% after 10 minutes. And with the 1+1 protocol of equilibration and vitrification solution for 1 minute each, that goes back up to 49.9%. But you can see with the dotted line, the intracellular osmolality is actually very similar in both those protocols. So that kind of opened up the idea of fast warming, that it could be done very quickly. And one of the earliest studies was by Mans et al. They were using Fujifilm Irvine kits, and they were using embryos that had been donated for research. So they had 32 embryos for standard warming, that you can see in the blue histograms, and 39 for ultra fast warming in those maroon histograms. They had a total, so that was the research step. So on the left, and you can see that pretty much there was comparable immediate survival and re-expansion by 24 hours. In the right-hand histograms, they looked at a total of 100 patients that were randomized between those two groups. So 51 patients had embryos warmed with the standard protocol, and 49 patients warmed with the one-step rehydration protocol of 1 minute, and then transferred to culture media. The average age between the two groups was not significantly different, so 35 and 36 years, not much, 35 and a half and 36 years. And the pregnancy rates between these two groups, you can see, although they were not significant, so they weren't significantly different, but nevertheless they were higher with the one-step warming. So 66.7% with one-step warming, with the standard warming, and 83.3% with the one-step warming. And likewise with the implantation rates, there was a trend, although not significant, towards a higher implantation rate with one-step warming. So the take-home message from this early study was that you could get at least as good, if not better, results with one-step rehydration, but probably just as importantly, you'd save over 14 minutes of operator time per warming. So for over 100, with 100 thaws, for example, you could save 1,400 minutes. So that's a significant saving of time. This very nice study by Jürgen Lieberman et al. Actually kind of demonstrated the proof of principle of this one-step warming protocol in some detail. So they were actually using 400 microliters for the warming, and this was a retrospective cohort study of the clinical outcomes of a large number of blastocyst transfers, so 833 transfers with one-step warming versus 2,606 transfers following standard multi-step warming. And again, they used Fuji Irvine vitrification media, and they were using high security vitrification straws. And as you can see in the table, the cryo survival rates were identical between the two protocols, with comparable clinical pregnancy rates. And even though, statistically at least, the patients were very slightly younger at the time of blastocyst warming and had slightly more embryos statistically transformed following the multi-step warming, the implantation and ongoing pregnancy rates with the one-step protocol were significantly higher than those in the multi-step protocol. And also the miscarriage rate was significantly lower. So this was a really encouraging finding with one-step rehydration. They also conducted a subgroup analysis to examine whether those protocol differences remained despite the use of pre-implantation genetic testing, or PGT, versus untested embryos that had been transferred. And they found that comparable outcomes persisted when assessing implantation rates in patients with euploid embryos in the one-step protocol versus the multi-step protocol groups, with a significant improvement of 5.8% being observed. And that was still present when compared with untested embryos, with a 6.6 difference being observed. Ongoing pregnancy rates were improved by a difference of 5.2% in PGT embryos in the one-step rehydration group versus the multi-step rehydration group. And similarly, the miscarriage rate in those PGT embryos was significantly improved, with a difference or a lowering of 3.4%. So overall, the one-step protocol improved outcomes in both patients having PGT and in patients receiving untested embryos, though the improvements were larger and more significant in those patients having euploid embryos transferred. And they did a final subgroup analysis looking at percentage differences on the day of development per protocol use. So this is the day when these, of course, when these blastocysts were vitrified, not when they were warmed. And significant differences were observed when comparing, again, the one-step rehydration group with the standard multi-step rehydration group on day 5 embryos in implantation rate, ongoing pregnancy rate, and miscarriage rate, with differences of 6.6%, 6.3%, and a lowering of 4.1% respectively. For day 6 embryos, a statistically significant difference was observed when comparing the one-step protocol with the multi-step protocol, but only in the implantation rate, with an 8.6% difference. And in day 7 embryos, no parameters demonstrated any significant difference. So it's obviously important, again, that you have good quality embryos that are developing at the appropriate time to see this benefit of one-step rehydration more so. But they did multivariate logistic regression analysis to control for things like patient age, biopsy, and PGT of blastocysts, and the day of blastocyst formation and expansion. And they found that rapid warming significantly increased the chance of an ongoing pregnancy by 26%. And that was really quite significant. But we have to remember that these blastocysts were collapsed prior to vitrification. And as I explained to you before, that is likely to help the speed of entry of these permeating cryoprotectants. But in addition to that, all these blastocysts underwent assisted hatching post-warming and prior to transfer. And these decisions were based on a previous study where they looked at various biopsy blastocysts. So another study, this Lemons et al. In this study, blastocysts were vitrified with rapid vit blastar kit and stored in liquid nitrogen, and these were not collapsed and not hatched post-warming. Again, they compared a standard protocol, their group 1, and compared that against a ultra-fast protocol group 2. But otherwise, the blastocysts were thought of the same kit. And of course, only one step used in that ultra-fast warming protocol. And then these blastocysts were moved into the conventional culture medium. And what they observed was that conservation of staining and spatial organization of cell fate markers following the ultra-fast rapid protocol were comparable, cryosurvival, re-expansion of the blastocele, clinical pregnancy and live birth rates, all these variables were comparable between the two rehydration protocols. So again, looking at this in a bit more detail, um, in terms of markers of viability, this was again very promising in terms of the rapid warming protocol. Um, what is our solution? The company I work for, well, we held back a little bit to see how things were going, but we've eventually brought out our own solution, which is the so-called viable Superfast Blastocyst warming kit, and there are six 2 ml vials of 1 molar sucrose warming solution in each of these kits. So up to 24 blastocysts can be warmed rapidly using 0.5 ml of that warming solution. So at the moment, it's identified as being for blastocysts only. There is no vitrification system specified, and as well as sucrose, there's 20 milligrams per ml of human serum albumin. There's MOPS buffer used, and that 0.5 ml is a minimum volume that is recommended, but of course users can opt for using larger volumes if they wish to. And there's no recommendation to do any washing prior to transfer into your standard culture medium. And as with many other media, it has a 7-day open shelf life. So what's the clinical evidence to support the use of this particular kit? So as you can see, using the viable Superfast warming kit, 1 minute protocol in the blue histograms versus the SAGE multi-step warming protocol. So these are both SAGE kits, of course. You can, in the maroon histograms, you can see that there's no significant difference in blastocyst cryosurvival, biochemical pregnancy rate, and clinical pregnancy rate. And indeed, between these two groups, there was no significant difference in maternal age. But the study was not controlled for embryo quality or for poor prognosis patients. But otherwise, there was no significant difference in the number of double embryo transfers, for example, between these two groups. So what else might you want to think about in terms of fast warming? What else has been reported that's relevant? Well, the three main things that I think are particularly interesting are universal warming, again, sucrose concentrations, bearing in mind that these are rapid warming kits, and the stages of embryo development that rapid or fast warming or one-step warming can be applied to. So the focus of this study, again by Lodo Parmigiani et al., was to assess the post-warming dilution and rehydration of embryos with a single step of 4 mils of non-permeable solute. And depending on the kit, this was either sucrose or trehalose. And this was, of course, again at 37 degrees C. And that was in order to elute out any cryoprotective agents rapidly prior to rehydration in isotonic media or culture media. So each patient's embryos were vitrified and warmed off-label with various combinations of commonly used vitrification kits, the mean female age and number, proportion, and indeed the morphokinetic quality of day 5 and day 6 embryos transferred was comparable for all study groups. And cryosurvival, clinical pregnancy, and implantation rates were also comparable for all study groups. And so again, it suggests that as well as with standard warming, with fast warming, again, universal fast warming seems to be applicable. There is, however, some variability in reports regarding the relative efficacy of different concentrations of sucrose used during one-step warming. Again, Jürgen Lieberman and colleagues, they did a study that suggests, and you can see that on the left here, that suggests that a 1 minute exposure to 1 molar, 0.5 molar, or even 0.25 molar sucrose results in comparable cryosurvival with a significantly higher blastocyst hatching rate using the 0.25 molar sucrose. Whereas if we look at another study by Emily Smith et al., they found that in fact 1 molar sucrose was better in their hands. So again, there are conflicting reports out there, and so we have to be cautious, I think, in trying to use lower concentrations of sucrose, and obviously stick to whatever the instructions for use are with the different one-step or fast warming kits that you happen to use. Now, what about cleavage stage embryos? So this was a preliminary study of 377 donated vitrified embryos from 210 patients. So there were 177 cleavage stage and 200 blastocysts. And these were randomly allocated and warmed, again, using either a standard or a one-step warming protocol, again, using Fujifilm Irvine vitrification warming kits. And this was followed up with culture with NACA one-step medium within a time-lapse incubator. And they found that the cryosurvival rates, again, were comparable, both between warming protocols and between cleavage stage embryos, which they actually got 100% cryosurvival, and blastocysts, where they got 99% cryosurvival. But they saw significantly higher morulation rates, and you can see that in the second set of histograms in that first chart, that achieved following the one-step warming in the lighter blue bars there, and otherwise expanded blastocyst formation was comparable between the two warming protocols. The time required for full re-expansion of the blastocyst, however, following the one-step warming of blastocysts, was significantly longer, which is quite interesting. But otherwise, there was no significant difference in the proportion of fully expanded blastocysts between those warming protocols. And I'll talk a little bit more about this later on when we consider the safety of these rapid warming approaches. So I said I would talk about post-warming recovery because this kind of is related, indirectly at least, to these differences between rapid and conventional warming, I believe, at least. So this was the first study that showed the importance of recovery culture. So this was a longitudinal study that analyzed clinical outcomes of blastocysts that had been cultured in NACA one-step medium, then vitrified, followed by warming and recovery culture in either NACA one-step or SAGE one-step GM-CSF medium, so containing granulocyte macrophage colony stimulating factor, which we know is a survival factor. So this recovery culture was for 3 to 5 hours post-warming and prior to transfer. And these blastocysts were also transferred in their respective recovery medium. And you can see in these histograms here that the warmed blastocysts provided with recovery culture and embryo transfer in SAGE one-step GM-CSF resulted in a significant increase in biochemical, clinical, ongoing pregnancy and live birth rates. And furthermore, the neonatal outcomes of the babies born following recovery culture of vitrified warm blastocysts in stage 1-step GM-CSF medium were no different to those following recovery culture in the NACA 1-step medium. Now, this has been replicated to some extent. This was a retrospective cohort study in 360 patients that had 575 cryotransfers at IVF Life in Alicante, Spain. And this data was presented at ESHRE last year. So in group 1, where they had 185 patients, they had post-warming culture for 2 to 4 hours and transfer in stage 1-step GM-CSF. And in group 2, with 390 patients, these patients had post-warming culture for 2 to 4 hours and transfer in continuous single culture complete medium or global total medium or embryo glue. And otherwise, donor oocyte and PGTA utilization were comparable between groups, and patient age and cycle history were controlled for in the analysis. And what they found was that the use of stage 1-step GM-CSF medium for recovery and transfer was associated with a 47% increased probability of a positive biochemical pregnancy, irrespective of the age and status of the patients, for example, if they had recurrent pregnancy loss. And that the use of that stage 1-step GM-CSF medium was also associated with a higher probability of clinical pregnancy and lower probability of miscarriage. And during that same ESHRE conference last year, Dr. Lanos Medrano presented further data comparing recovery culture and transfer of vitrified warm blastocysts, in this case from 1,580 patients. And as can be seen in this histogram, the histogram, the rates of clinical and ongoing pregnancy were significantly higher in those patients, again, whose vitrified warm blastocysts underwent recovery culture and frozen blastocyst transfer in the stage 1-step medium. And interestingly, when they did a sub-analysis of those patients that didn't have PGT versus those patients that had PGT-AA and therefore euploid embryos transferred. And what they found was that the difference between the warming with stage 1-step GM-CSF versus the controls was most significant in those patients not receiving transfers of blastocysts that were designated as euploid following PGTA. So one question we have to ask, of course, is whether 1-step warming is safe. And there have been various studies looking at this. So Chaplier et al. In 23 observed the same blastocyst diameter after 24 hours in culture and comparable rates of full hatching and no difference in expression of stress-related genes between standard and 1-step warming. De Gazelle et al. In 24 reported higher percentage of fully intact blastocysts post-warming using the ultra-fast 1-step method and a low number of damaged cells. And Jiang et al. Found the same live birth rates but lower multiple gestation rates and birth weights in the ultra-fast group. And Xu et al. Reported normal meiotic spindle integrity within oocytes. So as I said, I would come back to that Shoya et al. Study. And in that study, they also used time-lapse microscopy that revealed that the frequency of blastocele collapse, and you can see this in the histogram, there's that pair of bars on the right, comparing the standard warming and 1-step warming, you can see that the frequency of blastocele collapse was reduced from 50% to 30% following 1-step warming. whereas all other morphokinetic parameters were comparable between the two warming protocols. However, immunostaining with Hurst for live nuclei and propidium iodide for dead cells 3 hours after warming also showed that the proportion of viable cells was comparable between the standard and one-step warming, as were in fact the rates of blastocyst adhesion and the extent of trophoblast outgrowth. It's not all good news, though. This is one experimental study that did foreshadow caution. So in this study, 326 vitrified blastocysts donated for research were randomly allocated to conventional or shortened warming protocols. In this particular study, in the presence or absence of fatty acids during warming and the recovery period of 2 hours. And the degree of blastocyst volume recovery, as you can see, was significantly higher after the shortened warming. So you can see that in the red curve here. You can see that it goes above 100% to 111% at 120 seconds and doesn't return back to around 100% until about 300 seconds, compared with the standard warming where the blastocyst volume remains below 100%. So is this a concern? Well, it may be. Furthermore, they looked at cell membrane collapse, and they observed this within the trophectoderm during the shortened warming process. And you can see that in the micrograph in the lower panel on the right-hand side. You can see the arrow pointing to a trophectoderm cell collapse. But otherwise, blastocyst cryosurvival and expansion status following recovery culture were comparable among the different experimental groups. They also looked at apoptotic cells using annexin 5 staining, and also dead cells, as using the propidium iodide staining. And you can see again in that histogram on the right, with the significant stars on the bars there, that the proportion of necrotic cells, whether they were using their normal media or the fatty acid supplemented system with the shortened warming protocols, were significantly higher. So they recommended using conventional protocols with fatty acid supplementation. But otherwise, blastocyst adhesion rates were comparable, but trophoblast outgrowth was actually smaller in the short protocol. So this is one study that gives a counter argument to some extent against those other studies I've showed you. Okay, last but not least, let's quickly look at ultra-fast vitrification and warming. So combining the two. So this ASRM poster was a retrospective analysis of 754 FET cycles using 3 different groups, so conventional vitrification and warming, and they compared that with conventional vitrification with rapid warming or rapid elution of cryoprotectant, so that 1 minute warming. And they also compared it with ultra-fast vitrification. So this was a 2-minute cooling with equilibration solution combined with a 30 to 60 seconds cooling with vitrification solution. Again, combined with rapid elution. And what they found was no significant difference, as you can see, in these cryosurvival and biochemical pregnancy rates, but nevertheless a trend towards higher rates with reduced equilibration and rapid warming. So again, suggested that ultra-fast vitrification may be just as good. This study looked at conventional vitrification and dilution again, comparing with ultra-fast rapid vitrification and rapid elution or warming, and they used a range of different sources of oocytes for this research study. But overall, cryosurvival rates were significantly higher with the ultra-fast vitrification, rapid dilution compared with conventional vitrification and dilution. And in addition, they found that with all these types of rapid vitrification, that if they used artificial oocyte activation, they could get some parthenogenesis in these oocytes, and they actually got 4 blastocysts that were produced. Otherwise, meiotic spindle integrity and retardants didn't differ between these vitrification treatments, suggesting retention of cytoskeletal integrity and spindle formation. And lastly, this randomized prospective non-inferiority study, including fresh oocytes, both autologous and donated, that were discarded from ICSI cycles, compared ultra-fast vitrification with conventional and fast warming, and immediate survival and viability 3 hours after warming being evaluated. You can see no significant difference, but a trend towards the group 2 in the lighter blue bars with the fast vitrification. So in summary, that Gallardo et al. Paradigm shift study demonstrated that reduced times of exposure to cryoprotective agents in human oocytes and zygotes produces comparable outcomes. Traditional warming protocols tend to follow controlled stepwise rehydration procedures, but this is largely to mirror those that were developed for slow freezing and thawing systems. But nevertheless, one step or fast rehydration has been shown now to be effective in human blastocysts, provided that the warming rate is maximized. And it appears that fast warming may be effective with different concentrations of extracellular cryoprotectants, in this case sucrose. But 1 mole of sucrose may enable faster recovery. Over rehydration causing excessive swelling and membrane rupture may be a concern that we have to consider and keep an eye on. But otherwise, clinical outcome data are generally reassuring, and fast vitrification and fast warming certainly looks promising. That was of some use to you. I'll stop sharing, and I'll be happy to tackle any questions that you may have. I don't know whether I should look at any Q&A. No, it doesn't look like it. Yeah, so it's all exciting stuff, and it wouldn't surprise me if you have some questions.
Uh, Thank you so much, sir, for the wonderful session.
Welcome, Priya.
It was very enlightening. I was actually curious to know about the combination of the ultra fast vitrification of oocytes and its subsequent thawing, and it's very interesting to see that the group that had the 1 minute of warming in the DS solution while we skipped the TS solution, the 1 molar sucrose solution, it was very interesting that that group had higher results.
Yeah, so Mitch Schu's group has done most of the work in this area, along with Jürgen Lieberman, of course. Yes, it does seem that if we reduce the exposure of these oocytes, whether it be to the vitrification solutions or the warming solutions, that we're getting as good, if not better, results. Because the suggestion is, of course, that you're reducing the exposure to suboptimal temperature, suboptimal osmolality, and therefore osmotic stress. But also, we mustn't forget, we're also reducing the exposure of these cells to oxidative stress. So the shorter that we have these oocytes and embryos outside of the incubator, the better, I would imagine. So I think there's probably going to be a combination of factors that are combining to likely give better results. And now we know that these cells can survive such rapid vitrification and rapid warming. Then we can then hopefully avoid some of the other factors that we know may be detrimental.
True that. True that. I also wanted to just ask you that can we try the same protocol for embryos as well?
It seems
The
so.
ultra-fast
So,
vitrification for embryos as well.
oh well, one would hope so. I'm not sure that there is data looking at that. So the Shoya et al. Study that I showed you, that was on
Hmm.
ultrafast warming of cleavage stage embryos. So that certainly is promising, and these cleavage stage embryos that were subjected to ultra-fast warming compared with standard warming, they did as well, and they in fact
Right.
performed as well as blastocysts
Uh,
with ultra-fast warming. But I think it remains to be seen whether the same concept of fast vitrification works equally well with cleavage stage embryos. Again, we have to bear in mind that the cell volume uh, to surface area ratios are different in these different stage of embryo development. But I would be hopeful that just based on the
right.
Gallardo
Something worth
study. That
trying.
Yeah, I mean, if these cryoprotectants, these permeable cryoprotectants, if they reach the required
Right.
solute concentration in such a short time within oocytes, I really can't see
Mm-hmm.
why they
Yes.
shouldn't do likewise within blastomeres in cleavage stage embryos.
A lot of people are still skeptical about using it, but I
Okay. Well,
yeah,
that
but it has
skepticism is
given good
good.
results. I have tried a few of those, and I also tried the rapid freezing of oocytes, and surprisingly, I did not expect to get such nice plump oocytes upon warming them.
That's
So
good.
yeah, that's there.
Yes.
Also, the forum is open to questions, and if anyone wants to know anything about the session today, to please drop your questions in the chat box.
Yeah, I'm looking in the chat box. So don't be shy. If you have any questions, please. There's no such thing as a silly question.
Exactly.
I think this session, this is predominated by our students. They are mature
Okay.
students. Yeah. So
Well, that's
still
good.
they have a long journey to go, and they are in the process of, acquiring the embryology skills and embryology chapters. So probably that's why we have less questions this time. In fact, there's no question. Then
Well,
maybe
I hope I
Priyanka,
haven't shell shocked them with all the data. But yeah, I mean, in many respects, it's an easy transition, of course, to go from conventional warming and even, I would imagine, from conventional vitrification to ultra fast or one-step warming and fast two-step vitrification. So I think, like you say, Prieto, it's a matter of
Yes.
proving to yourself in your hands in a
Yes.
controlled group of hopefully research material initially,
True, true. Right.
that you're to give you the confidence that
Yes, I think
these
every
these
lab, every
embryos and oocytes
embryologist
survive.
should try to do the protocol once, see for themselves what they think, and then proceed accordingly. And
Yeah,
definitely the bonus point is it takes down the time from 15 minutes to 1 or 2 minutes. That's
yeah.
absolutely amazing for labs where we have to do multiple freezings or multiple thawings at one time, which usually becomes hectic for one person. So this protocol is going to be a great, great help for those people. Exactly.
Yes, and we used to argue whether in terms of the relative time, whether if we had a lot of oocytes or embryos to vitrify, whether it was actually more efficacious than slow freezing. So now that you can actually reduce that time even further, so to a really
Definitely.
quick process, I can imagine once people are convinced that it works in their hands, there's no going
Yes.
back, is there really?
No, no, no.
No,
Obviously, that's
you
going to be
you
a
wouldn't
great
want to go
thing.
back to spending all that time
I would
doing that
always keep
vitrification and
following
that
this.
warming.
Yeah,
Yeah,
if I get to try it properly in the lab, like in a full-fledged working lab, then I would definitely opt for this technique.
yeah,
But that's there. We need more studies. We need more research happening
we
so
do.
that everyone can be sure that they need to follow this protocol and how to follow it. That's it. Otherwise, it's going to be completely time saving and less stressful for all
Yeah,
embryologists. So that,
I agree with you entirely. We always need more data.
yes,
And of course, we have no long-term data.
yeah,
Um, so yeah, all this, the data will be very important to keep an eye on the safety and reliability of these rapid techniques. But all
yes,
we can say is that so far so good. I think apart from the one article which suggests caution, which is sensible,
true.
I think most of the data is actually quite reassuring, quite
Yeah, yeah.
encouraging.
That's what I was looking, and I was actually feeling very good about it. That the data looks
Mm,
really promising.
yeah.
So yeah, definitely worth trying. Uh, So, since there are no questions today, I think it is best that we can close the session.
Okay.
So, again, thank you so much, sir, for the wonderful session. It was very interesting, and I was very, looking forward to hearing from you. It has
My
been
pleasure
always a
as
pleasure
always.
from all your sessions. So, thank you so much, and thank you everyone for being here today with us. Uh, we'll meet you again next month on the third Friday, and with a new topic, with a new speaker. And that's all. Thank you. Thank you. Have a great day.
Okay.
Thank you,
Thank
Steve.
you.
Thank you.
Bye then.
Thank you.